The first organelle that we should consider is the nucleus, which is also considered the control center of the cell. The nucleus is the control center because it stores the hereditary information, or the DNA, the deoxyribonucleic acid. This DNA will come in two forms. Number one, it could be in the form of chromosomes, or number two, it could be in the form of chromatin. When it's in the form of chromosomes, it's going to go ahead and be specifically in that form when the cell is actually dividing. However, when the cell is not dividing and it's at rest, it's in the form of what's called chromatin, or uncondensed DNA. So chromosomes are going to be condensed, tightly wound DNA, whereas chromatin is going to be uncondensed DNA that in essence looks like spaghetti. If we go ahead and we take a look at this diagram right here, what we see is a cell. And in the middle of that cell, we see that large purple structure. And again, that purple structure is the nucleus. In the very middle of the nucleus, we're going to see a structure called the nucleolus. The nucleolus is very important because it's in that structure that we're going to go ahead and have the assembly of ribosomes. Ribosomes are going to be small organelles that are very important for the production of proteins. Now, you're going to find your DNA inside the nucleus. But to go ahead and make these proteins, the genetic information has to get from the inside of the nucleus out. And it's going to go ahead and leave the nucleus as a copy rather than DNA called RNA, and it's specifically going to leave through nuclear pores. Notice that when RNA leaves the nucleus, or the copy of DNA leaves the nucleus, it's going to be passing through these nuclear pores, and the nuclear pores are going to go ahead and be found at the juncture of an inner membrane and an outer membrane surrounding the nucleus. And this inner membrane and the outer membrane are going to go ahead and compose what is called the nuclear envelope. Outside of the nucleus, you're going to have something that's considered the endomembrane system. The endomembrane system is simply an extensive system of interior membranes that divides the cell into compartments. And the endomembrane system will go ahead and consist of, number one, the endoplasmic reticulum, number two, the Golgi complex, number three, structures called lysosomes, and number four, structures called peroxisomes. First, you have the endoplasmic reticulum, and this is going to be that structure that surrounds the nucleus. The endoplasmic reticulum, or the ER for short, is going to go ahead and be an internal membrane system composed of channels and membrane-bound vesicles that are going to go ahead and break off from those channels. You have two distinct regions of your ER. And you're going to have, number one, your rough ER and your smooth ER. The rough ER is going to be studded with ribosomes, and that's specifically why this particular type of ER is called rough. And the rough ER is going to be involved specifically in protein synthesis. Again, the ribosomes are important for protein synthesis, hence the rough ER is going to go ahead and create proteins. The smooth ER, on the other hand, is not studded with ribosomes. Therefore, it's going to be involved with lipid and carbohydrate synthesis. So again, keep in mind, in general, this ER, or endoplasmic reticulum, is involved in the production of macromolecules. The diagram that you see is going to go ahead and indicate for you both the rough ER that's studded with ribosomes in blue and the smooth ER that is not. If you look at the micrograph to the right that was created by an electron microscope, you can go ahead and see the rough ER and the smooth ER. In addition, you should realize that macromolecules that are created by the ER are going to go ahead and have vesicles leave the ER and transport those materials to the Golgi complex. The Golgi complex is going to be composed of several Golgi bodies. These are going to be flattened membranes in the cytoplasm, and they're stacked again into what is called the Golgi complex. I consider the Golgi complex to basically be the FedEx of the cell, in so much as it's going to go ahead and pick up stuff from the ER and transport to new locations within the cell. The Golgi complex will collect, package, and modify and distribute molecules within the cell. In this particular diagram, you're going to see the ER that is going to be surrounding the nucleus, and below that ER, you're going to have vesicles being given off and sent specifically to the Golgi in pink. You're going to have transport vesicles coming into the Golgi complex and secretory vesicles leaving the Golgi complex, again carrying macromolecules. And if you go ahead and look to the right, again you're going to see an electron micrograph that indicates for you the Golgi complex and vesicles surrounding it. Next we have lysosomes, and lysosomes are again part of this endomembrane complex. And these lysosomes ultimately are going to arise from the Golgi complex. And if you look at the micrograph to the right, you're going to see examples of lysosomes. Lysosomes are important. They actually contain enzymes that break down macromolecules. They function specifically in the intracellular digestion of worn out cellular components and also substances that are taken into cells. And the resulting material from them is going to ultimately be recycled, and it can be reused in the creation of new macromolecules. We also have as part of this system peroxisomes. Peroxisomes in this particular case are going to go ahead and be indicated by the black dots in the micrograph to the right. Now, while your lysosomes are going to arise from the Golgi, your peroxisomes are actually arising from the endoplasmic reticulum. And the peroxisomes are going to contain two sets of enzymes. One set specifically is found in plants, and it's going to convert fats specifically into sugars. And the other set is found in animals, and it will go ahead and detoxify various harmful molecules. The ER and the Golgi complex function together as a transport system in the cell. If you look at this diagram that indicates specifically how the endomembrane system works, you're going to see both the ER and the Golgi. The ER is going to be indicated again in yellow. In yellow, you're going to have your smooth ER and your rough ER that's studded with your blue ribosomes. Again, your ER is going to go ahead and create macromolecules. Vesicles will be given off by the smooth ER and sent to the Golgi, which is indicated in this salmon color. And you're going to go ahead and have macromolecules packaged and redistributed from the Golgi to different areas within the cell. Keep in mind that you're going to have lysosomes come specifically from the Golgi. And also keep in mind that some of the things that are produced in the cell actually are going to go ahead and leave the cell and perform other functions throughout the body. So it's important that you have production of vesicles that ultimately will go ahead and fuse with the plasma membrane, releasing the things that they actually contain. And once those things are released, they can travel to new locations in the body and perform the function that they were meant to perform. Now, there are some organelles that actually contain DNA. Remember I said the nucleus contains DNA of the cell, but there are some organelles that are special. And these organelles found in eukaryotic cells specifically are going to contain DNA of their own. According to the theory of endosymbiosis, organelles were derived from ancient bacteria that were assimilated by the eukaryotic cell. In the figure to the right, you see what represents eukaryotic cells taking up bacteria, ancient bacteria in green. And then what you have here is a situation where you have membranes surrounding that bacteria, and that bacteria comes to live in and exist in the eukaryotic cell. This bacteria would have had its own DNA. Hence, that is likely why we see that some of our membranous organelles have their own DNA, which is different from the DNA you find specifically within the nucleus. The two organelles that represent those organelles that have their own DNA include number 1, mitochondria, and number 2, chloroplasts. Mitochondria are found in almost all eukaryotes, and chloroplasts are found specifically in plants and algae. Now, it's in the mitochondria and the chloroplasts that you find DNA that is different from the DNA specifically found within the nucleus of every single cell. And again, it's suspected that the DNA found in the mitochondria and the chloroplast ultimately resulted from the fact that the mitochondria and the chloroplast at one point in time, a very long time ago, were likely bacteria that were taken up by cells and incorporated into them. The mitochondria are the powerhouses of the cell. They are going to extract energy from organic molecules through what is called oxidative metabolism. These organelles are going to be sausage-shaped organelles, and they're actually also about the size of a bacterial cell. And if you go ahead and you look to the right, you'll see a micrograph created by an electron microscope of a mitochondrion. Like bacteria, mitochondria also possess circular DNA and also divide by simple fission. Here we see an example of a mitochondrion, or mitochondria for plural. What we see here are various membranes and intermembrane spaces. Number 1, we have an outer membrane. That outer membrane is then going to go ahead and be divided from an inner membrane by what is called the intermembrane space, or between the two membranes. Within the inner membrane, what we have is an area referred to as the matrix. And we also have structures referred to as crista, or cristae for plural, that are going to go ahead and provide significant surface area for the inner membrane. Now, you're going to go ahead and find that within the matrix, you're going to find the mitochondrial DNA. And also, these crista, or cristae for plural, will significantly increase the surface area of the inner membrane, which will be extremely important in the production of energy for a cell. Next, we have our chloroplasts. And the chloroplasts are going to go ahead and be the energy capturing centers of the cell. While the mitochondria are going to go ahead and make the energy, they're going to actually make ATP, chloroplasts ultimately are going to go ahead and capture energy from the sun in the process of photosynthesis. This is going to be performed by plants and by algae. If you look specifically at the micrograph to the right, it is going to be a micrograph of the light microscope. you're going to go ahead and see several cells. And within each cell, you're going to see several small, round chloroplasts. These chloroplasts are going to go ahead and capture the sun's energy and turn it into a form called glucose, which is a sugar. Chloroplasts, like bacteria, possess circular DNA and also divide by simple fission, again providing support to the theory of endosymbiosis. Chloroplasts also are surrounded by two membranes. However, the inner membrane is much more complex than the outer membrane. Here we see a diagram of chloroplasts. What you have to the left is the typical plant cell, and what you can see in the plant cell is an example of 3 chloroplasts. If you blow one of those chloroplasts up in the diagram, you're going to have an outer membrane. You're also going to have an inner membrane. And that inner membrane is extremely complex, and it's going to be indicated specifically by the green area. What you have here are going to go ahead and be, number 1, several small penny-shaped structures called thylakoids. These thylakoids can be stacked together as something called a granum. So hence, you're going to have ultimately several grana. And these structures are going to be floating in what's called the stroma, which is akin to the cytoplasm of the cell. And it's going to go ahead and be this inner membrane that's going to be extremely important for capturing the energy of the sun. And you can see here that you have a lot of surface area for this inner membrane that will allow maximum capturing of the sunlight's energy. The figure to the far right is going to be a micrograph of the chloroplast created by an electron microscope. And it's those thylakoids that are going to go ahead and be those specific sites of photosynthesis. The thylakoids, which are stacked together as the grana. Next, we have the cytoskeleton. This is going to represent the interior framework of the cell. The cytoskeleton is a dense network of protein fibers that, number 1, support the shape of the cell, and number 2, anchor organelles. You can go ahead and see in this particular diagram to the right, organelles being anchored by the cytoskeleton. For instance, you see both ER at the top of the cell and mitochondria toward the bottom of the cell. They're being anchored by the cytoskeleton. There are 3 different types of protein fibers that are going to go ahead and compose the cytoskeleton. Number 1, you're going to have microfilaments indicated by the green filaments toward the bottom of the cell. Number 2, you're going to have microtubules indicated by the blue-green filaments that run the length of the cell. And number 3, you're also going to have intermediate filaments that are represented by the lattice work-like structures that are in brown. Again, you have 3 different kinds of protein structures or protein fibers within your cytoskeleton. Number 1, you have intermediate filaments. Your intermediate filaments, indicated in brown below, are going to go ahead and basically be thick ropes of intertwined proteins, again, that are going to play an important role in securing organelles in place. Next, you're going to have your microtubules, and these are going to be hollow tubes made up of the protein tubulin, and again, you can see them running the distance of the cell. And finally, you're going to have your microfilaments, indicated in the light green color toward the bottom of the cell. And these are going to be long filaments that are made up of the protein called actin. Centrioles, remember, are going to be important structures in animal cells, and they're going to be responsible for anchoring and assembling microtubules. What we have here, for instance, is a cell, a typical animal cell, and within that cell you're going to go ahead and see centrioles. These centrioles are going to go ahead and be made up of something called microtubule triplets. You can see 3 microtubules grouped together as a triplet, and the centriole itself is made up of several of them. In addition to animal cells, these are also found in protists. And these too may have originated as symbiotic bacteria, just as mitochondria and chloroplasts did. Now, cell motion is tied to the movement of microfilaments and microtubules. So not only is the cytoskeleton important for securing organelles in place, but it's also extremely important for cell movement. Changes in microfilament shape permit cells to change their own shape very quickly and also crawl or swim. They also are used in the division of animal cells. Organelles used in cell movement include, number 1, the flagella. These are typically going to be structures that are long and few in number, and also cilia. These are going to be structures that are typically short and numerous in number. Right here we have an example of a single-celled organism, a protist called the paramecium, and we have on that structure, or on that cell, several cilia, multiple cilia that are going to be extremely important for the movement of this organism, and also the cilia are going to allow the organism to go ahead and move food particles toward its mouth. Let's go ahead and take a closer look at the structure of flagella and cilia. In this particular diagram, we have an example of the eukaryotic flagellum. The bottom of this particular figure represents the cell body, and emanating from it, we're going to have a eukaryotic flagellum that might be important for movement of the cell. And what we're going to have here is a situation whereby in the body of the cell, you're going to have what's referred to as a basal body. The basal body, as you can see here, has several microtubules arranged in triplets. You're going to go ahead and have several of them, and this basal body is going to go ahead and serve as an anchor to the flagellum itself. The flagellum itself, however, is going to have a slightly different arrangement of microtubules. Rather than the basal body, which is going to have triplets, the flagellum itself is going to have what's called a 9+2 arrangement. It's going to go ahead and have its microtubules arranged in doublets. 9 doublets, which are going to go ahead and surround one doublet. 9 pairs surrounding 2 microtubules in the center. Again, it's going to be this flagellum that's going to allow a cell to actually move. Remember, the flagellum is going to be a whip-like structure that will be used to propel a cell forward. Microtubules are not only important in moving cells themselves, but also moving material within a cell. Microtubules will transport material within the cell over long distances. Please notice to the right that you have a large green microtubule. Again, this thing is going to go ahead and run the distance of a cell. And attached to that microtubule, you're going to have something called a motor protein, indicated in blue. There are 2 main types of motor proteins that could be attached to the microtubule. Number 1, something called kinesin, and number 2, something called dynein. Kinesin is a motor protein that moves vesicles to the cell's periphery. So kinesin is taking stuff toward the periphery of the cell because ultimately it's likely going to go ahead and want to take whatever is in the vesicle to the outside of the cell. Dynein, on the other hand, is a motor protein that moves vesicles to the cell's interior. So likely, when you have dynein attached to a microtubule, it's because something is coming into the cell. If you look at the diagram, again, you're going to see the microtubule attached to the blue motor protein, which is either kinesin or dynein, and then it's going to go ahead and be attached through a series of other proteins to a vesicle. Remember, this vesicle is likely going to go ahead and be coming from the Golgi complex. It's going to go ahead and likely contain things, macromolecules, that need to go somewhere so that they can be used for some particular process. The way that I like to remember kinesin from dynein is that with dynein, what you have is a situation where stuff is going downtown. Dynein, downtown. Stuff is going to be moving toward the interior of the cell. Remember that when you're dealing with a downtown, it's typically in the interior of a given city. And so in this particular case, dynein downtown toward the interior. Next, we have vacuoles. Remember I said that vacuoles can play an important role in plant cells. Number one, they're capable of storing dissolved substances, and they also can increase the cell surface area. Here we have a plant cell, and what we see here is an electron micrograph below it, where you actually have a very large central vacuole. You can see the plant cell wall surrounding the exterior of the cell. You can also see several chloroplasts within it. But again, that large clear area inside of the cell is the central vacuole. In protists, you also have something called contractile vacuoles, which are similar to your vacuoles in plant cells. The contractile vacuoles are also going to be used to pump excess water out of the cell as the cell takes on water. So what's going on outside the plasma membrane? Well, as I mentioned, you can go ahead and have something called cell walls. Cell walls will offer protection and support in plant cells, fungal cells, and protist cells, but cell walls are not found in animal cells. Fungal cell walls are made up of a structure called chitin, whereas plant cell walls are made up of a structure called cellulose. The diagram that we see to the right is going to indicate or illustrate for us the cell wall. What we have here basically are two cells. Up in the upper right-hand corner we have cell 1, and the lower left-hand corner we have cell 2. Surrounding those cells, we're of course going to have a plasma membrane, but then we're also going to have the cell wall, which is going to go ahead and impart structural support to the cell. You can see here that we're going to have what are called primary walls as well as secondary walls. And what we have in between the cell walls is something called the middle lamella, which acts as a glue so that the cells will stick together. We also have, outside of the plasma membrane, something called the extracellular matrix. The extracellular matrix is going to be a series of glycoproteins secreted by animal cells. These glycoproteins are going to go ahead and include proteoglycan, elastin, and collagen. And these different types of proteins are going to go ahead and coordinate behavior of cells in a given type of tissue. Collagen and elastin specifically are going to go ahead and provide protection to the outside of the cell. Fibronectin, on the other hand, is going to go ahead and connect the extracellular matrix to the plasma membrane and to proteins specifically that extend through the cytoplasm. Notice in the diagram to the right that you're going to go ahead and have large purple proteins called integrins that span the plasma membrane. And outside of the plasma membrane, you're going to go ahead and have that integrin attached to fibronectin. And that same integrin protein is going to go ahead and be connected to actin filaments on the inside of the plasma membrane, inside of the cell. And so in this particular case, you can go ahead and relay information from the outside of the cell to the inside of the cell.